Welcome, everyone. I'm Mario Franceschini, the president of the SfNIRS Society, and it's my honor to officially open the sixth biennial in-person fNIRS conference. It's really overwhelming to see you all here. After all the delay and the pandemic and all things that happened in the last few years, it's just feel like a dream, but it's real. So I'm very happy. So, so let me decide what to say. So yeah, it's been really great to see everybody again, and I'm sure it will be a few great days that we will share together. And we are broadcasting in Zoom because there is more than 100 people that couldn't come because of visa and other things. So hi guys, and hopefully next time we will see you too. So the society during this last 4 years has done a lot to keep us all united, to allow scientific and social interaction among members, thanks to Zoom meetings, Gather Town, monthly webinar organized by Rickson, educational tutorial by Judy Gervain, and the last year virtual meeting was just great. But so when we planned this meeting in 2018, we were not thinking about hybrid. We were expecting more than 500 participants to come to this venue. And here, we almost filled up the venue. So we have more than 400 participants, and it's awesome. I really didn't expect that. So I want to start with the slides. Oh, cool, it starts. So back in 2018, we made this Boston layout skyline, and in 4 years, Boston has grown like crazy. There are so many new high-rise buildings. But I'm so pleased to see that our society is also growing up at a similar rate. So we have an exponential rate of publications in the last few years. The society is thriving, and thanks to many new young scientists that offered their time and help making meetings like this one a success without having the crazy high event tickets like some bigger society. What I hope to see in the next few years is a growth in the sense of community, not just when we see each other, but also when we review NIH grants. So maybe this is just talking to the American audience, but I should apply to everyone. So I sit in many study sections, and what I saw in other fields, people fighting for their field. And I haven't seen that in our community. And I pledge you guys, be nice at the review section, because it come back to you. And you shouldn't be nitpicking. If there is something wrong, sure. But there is no enemy here, it's a community. We are all members of the same society, and more money goes to a person, it goes to the society and made the society grow. So think about that, and I think it's very important. So what else? Let me keep going. And so I'm so happy the conference is happening. The attendance is behind all my expectation, and I think we are ready to start. And can I take a selfie? Yeah. Okay, guys. Ready? I'm not that good, but oh, this is awesome. I love it. Okay, I take two. Yeah. So this conference was supposed to happen 2 years ago, and back then Claire was the president, and she would have given this welcoming speech. And then I will have follow as the conference chair. The pandemic messed up this plan. So I have to introduce myself, and the whole year, last year, I've been wearing two hats, but I don't wear hat. So when I talk to you as a president, I'm wearing a pink jacket. And not because I like pink, but because I think there should be more president wearing pink. And this goes straight to Joe. We want to see you in pink and for you, 2 years, whatever. And so now I'm passing the stuff to the chair. And I'll be wearing whatever color I feel like it. So, okay, back to the many color. So, okay, this is fun. So we started in 2018, I said, and so this didn't help because we planned everything, then we had to replan everything and replan everything. And the work was crazy. So we never done an hybrid conference before. And it's not just doing the in-person and the virtual, you have to merge them. And it's exponentially more difficult. Plus, we had no idea what to expect. We didn't know how many abstracts we get. We just had a virtual meeting last year, very successful. We didn't know how COVID pandemic was going. We didn't know how many people would have come here or not. So everything was kind of, let's wait a little bit longer, let's wait a little bit longer. And at the end, we were so crazy, overwhelmed by everything because we got 350 abstracts. That is more than what we ever got before. We got 520 registrants, 530. That is, in Tokyo, there were 430. So it was challenging. So yesterday I said, okay, let's see how many email conversation with the subject fNIRS 2022 I had in the last few months. So conversation, no email. So I had, in the last 6 months, 1,800 email conversation and more than 900 last month. So it's like my life stopped and I had to do the conference. But thank God it was not just me. I had a program chair here, Pepe Dan, that helped a lot with the program, and you have to thank him for the great selection. Then we saw that the hybrid component needed a virtual component. And while we had not done many abstracts to start with, we had 29 abstracts that were only virtual, they kept growing. So we ended up with 40. And we wanted to highlight them too. So we have a new chair position, is the virtual program chair. And Sabrina Brigadoi, I don't know if she arrived, but she was perfect. I don't know how to thank her. She saved us. And then there was the educational that in 2018, I said, let's do something more fun, more complex, more things. And thank God I had Miriam because it was really complex. I know it went very well, but it was a lot of work. And then, of course, Stacy, that we pay for a conference into a year and she had to work 4 years. So, and she worked a lot. So I want to thank all of them. And, but it's not just them. So it's all these young faces here that did so much work. So I want to name all of them. We start with Andrew. Hi, Andrew. He helped with the program. He helped with the Zoom session, and he's going to help with the Zoom here. It's awesome. Then we have Fen. Fen, oh my God. She's the master of Gathertown. If you've been there, she's the one that built all these little details, all the flower, the piano, the various room, is all her. And it's not just that, she had to make the poster room, it's more than 300 posters. And so because everybody was late and we made the deadline the latest as possible before we start Gather Town. What happened is the weekend before Monday, October 3rd, we had all this help getting the poster from the Dropbox and putting them in Gather Town. And despite me emailing, say, has to be an image, has to be JPEG, has to be less than 3 megabytes, there were all sorts of PDF, PowerPoint, 50 megabyte files. So they not just put the poster there, they had to reformat many of them and chase people that were missing. Plus, several of them, they help with other chores for the conference, like Anita here, Zara. She is having this awful job in the next few days, chasing speaker to get the talks inside the Dropbox. So they are in this computer and they are in Zoom. So please, if you're a speaker, for tomorrow, we have 20 speaker and we have 8 talk in the Dropbox. So she will email you all and make sure that these talks are in this computer. And then you go up there in the AV room and you can check that everything is okay. And if tomorrow morning you want to change a slide, you go up there and upload it up there. Don't give me 10 copies of the same, because then the chair will be, okay, which one I start? It will be confusing. Then Bryce is sitting over there, is the Zoom moderator. So he's the one that talked to the 120 people that you don't see but are here. And if they have any issue, they go through him. And so thank you, Bryce. Then I have Sam. Sam, you must have seen some of the great video he made with some of the speakers. He's responsible to make sure that all these talks, next few days, will be recorded and he will upload them in our webpage. So people that are virtual and they are in China, right now it's middle of the night, they can see at their convenience in a few weeks. And okay, Yuan, she helped with educational courses. She helped Miriam to make everything smooth with the last 2 day courses. Merdad, thank you, Merdad. So what he did was we automated, David Boss, he automated the website as much as he could, but there was a problem. Either we make the pages available for everybody, people that are member, people that are non member, people that don't pay, or we add the subscription to the fNIRS 2022 conference. And there was no way to do it automatically. So how many people there are here? He automatically added the fNIRS 2022 subscription to all of you. And finally, I have Guy and Luisa that they were taking care of the Facebook and Twitter, social media. They advertise everything and posted everything that's going to happen, all the news. So thank you a lot, all of you. So, but now let's move to the program. The program was huge. We started, we started making them in person, but then we had we wanted to start having people to look at the poster before, because we have only 2 hours and there is also lunch happening in the poster. So it was important to have people to give them the ability to go see the poster before that. So we started a week earlier. We had Gather Town poster. We had poster blitz. We also had a poster session for the virtual only posters. And everything went well. Fan told me that there were about 500 entry in Gather Town per day. So I call it a success. So, but let's focus now. Now is the in-person program. So we have a keynote lecturer, Joy Hirsch, that will come next. We have Bruce Tromber, NIBIB director, coming tomorrow morning. And we have 8 invited speakers. There is a special session in ergonomic with 5 speakers and a panel. And there are 46 contributor oral. So probably 12 are uploaded right now. So we need more. And then there are the 3 poster sessions with about 250 posters divided in 3 days. So for the poster, let me tell you now so I don't forget. There is this famous number PS1, that means poster session 1. Then there is poster session 2, PS2. And then there is a number that goes from 1 to 90. In that room where the posters are, there is not written PS1 or PS2. There is written 1, 2, 3, 4, 5. So you look at your number after the PS1 tomorrow morning and put your poster in that number. And then you take it out at the end of the day, and the next day, the PS2, put the next number, PS2, 0, 1, 2, 3, 4, in the poster board there. And please take it out at the end of the day because we have poster the day after. So hopefully everything worked great. So the venue is, we are at BU. I don't work at BU, but my husband, David Boas, does. So we got this venue because he can host a lot of people, and because back then we thought we will have a lot of people, and actually we have a lot of people. So this is the McCall Hall where there will be all the oral session. I told you, if you're a speaker, go check with the AV up there. Here is where we will have all our lunches, next door there. This is the poster, coffee breaks, and sponsor room. And there is tonight, if it's not too cold, we have the reception outside. It's beautiful. We can see the Charles River. There is thorough drive between us and river. There is an overpass. Don't think of crossing that street. And if it's cold, we can go back in and use that room. So Stacy told me to remind you guys to wear the badge. If you don't have the badge, we can tell you, please go out. So only people with the badges should come here. What else? Bathrooms are here. The Wi-Fi is simple, is guest, and there is no password, nothing, is very unencrypted. Okay. Social event on Tuesday. We go to the Royale. It's an historical theater, let's say club. We will have a lot of great surprises. So if you haven't bought the tickets, I know Stacy has a few more, so run to her. You can take Uber or train, or you can walk through whole Boston. There's a beautiful walk to get there. We end here at 6 and that starts at 7:30, so you have plenty of time to do that walk if the weather is decent. I think it's decent. So we know there is COVID, even if we don't want it. So please, you can stay without mask, but if you feel a little sick, wear a mask. If you had contact with somebody that is positive, have a mask. If you have COVID-19 symptoms, don't come. Do a test first, make sure you're negative. You can look at the conference in Zoom. This is really very important for the safety of everyone. For the virtual attendee. So you can find the Zoom link the same way. Okay, if they're here, they know that. But anyway, you can find it at the usual place where Gather Town is, plus I blasted everybody yesterday. So Bryce will kick out people that don't put their name on the Zoom. We want to know who's attending. The Q&A is disabled. So David created this beautiful page where each abstract has its own page, and we can add commenting in there and start a discussion with the presenter. You have to subscribe so you know when somebody asks you a question. So subscribe to your abstract and ask. If you don't have time to ask questions to the oral because the program is packed, go to the web and ask a question there, and people will respond. We were thinking of having a prize on the poster that has the most discussion, scientific discussion. So, and I think Guy and Luisa will take care of it. So there is the Zoom chat. Don't put question there because we will not see them. We will not answer them. And contact Bryce if you have any issues. So everything is recorded. Every speaker that speak here is supposed to be recorded. If you don't want to be recorded, you let us know. We stop the recording and start after your talk. But you have to tell us. And the recording will be saved and available on our website. So I talked too much. We move to introducing the keynote of tonight, and Pepe will do the introduction.
Okay.
Thank you. Oh,
Okay.
don't do that. Maybe it's me.
Okay, maybe.
Maybe.
Is it okay?
Yeah.
Okay. Hi. Good evening, my colleagues. Welcome to Boston. And before we begin, well, she made a really great introduction here, but well, we have to first thank Mari for arranging such a nice conference. Thank you very much, Mari. Yeah. Okay, now I guess you are enjoying the best season in the very beautiful city, Boston. And we are glad that we finally come here after the hard time we have suffered together. And it's great that we can have the social interaction here more than ever. So this is my great pleasure to introduce Professor Joy Hirsch at the Yale University and at UCL. Who has been the leading science in the social interaction study using functional NIRS in recent years. Let me give you a brief introduction of her. Well, just a moment. It's a really complicated one. So she graduated from University of Oregon, majoring in life sciences, and got a master's degree afterwards in psychology at Portland State University. Then she finished her PhD in psychology at Columbia University. Well, the combination of life sciences and psychology made her a really excellent background to continue her study. Then she then underwent lots of important academic positions, including there are a lot, actually. To make it short, she got an assistant professor and associate professorship at the Yale University School of Medicine. Then she became a professor at Cornell University, and after that, she became a professor at Columbia University. And if you would like to have the whole history of her, and you can visit her website at Yale University, and there you can find a detailed CV of her. So it's really surprising. You would like, I would like you to do that after the talk. Okay, and she is now a professor of psychiatry and neurology at the Yale University School of Medicine. At the same time, she is a professor of neuroscience at the University of College London. Okay, and I would say it would be really surprising to tell you that she is relatively new in the functional NIRS community. Rather, she used to work on the functional MRI research field for a long time. And she was established well in the studying the cortical network of human cognition in the late 90s and early 2000. And around 2010, she was really successful in studying functional MRI network study on conflict resolution. That's really great study too. And then there came a valuable moment to us. And in the middle of 2010, she became interested in functional NIRS to study social interaction. What is really good about her is that usually when people do the functional MRI research, and it's a usual path from coming from the functional NIRS to functional MRI, but they never come back. Sometimes, unfortunately, but she followed the opposite way. Great. She came from the field of functional MRI to go back to NIRS, and well, she became she became interested in doing experiments in the social interaction in the very natural setting. And anyway, welcome to the Functional Neuro Society. Great. Anyway, so created I've got to mention that too. And she created a great laboratory, which I should stress, with great scientists, including Shanjan and Adam Noah. Well, I've got to thank them for their really great contribution to this field. And thanks for hiring them, along with your colleagues, other colleagues. Also, it is really great to know that she really appreciates social interaction, and she has invited her friends and cousins, and also her husband, James Rossman, our other guest. And I'm really glad to find that we got a Nobel Prize laureate as an audience, not as a speaker here. Okay. Anyway. She has published more than 150 papers and attracted more than 20,000 citations. And she is now serving as on the board of directors and the chair of diversity committee of our society. So let me introduce this great scientist, the mother of hyperscanning, Joy Hirsch.
All righty. Okay. Thank you. It is such a pleasure to be here. Thank you, Pepe, for the amazing introduction. I hardly knew who you were talking about. Thank you. The most important thing he said is it's been a long journey. So here we are. This is truly a celebration tonight. It's a celebration of all of us being here. I mean, how long has it been since we have all been at a meeting together? My goodness. It's been 4 years, as Maury pointed out, since our society has met. And it is truly an amazing accomplishment that we are all here. So thank you all for coming to this meeting. And we have a couple of other thank yous before we get started. The first one, we haven't said enough about Maury yet. Maury, from the bottom of our hearts, from every language that we represent in this society, from every country that we represent, we thank you. From the bottom of our heart, we thank you, because this, guys, would not have happened without Maury. Maury has the award, I think, for the fastest response to any email that you send. At any time of the day, at any night, weekend, day, night, whatever it is, Maury will respond. So thank you. Maury had some friends. We have a board of directors that has managed this society since its very beginning. And this society is a very significant, a significant thing about this society is that it is managed not by professional people, but by the scientists who actually are the ones that build the science. So it's the scientists that are running things. And so I guess that's why it runs so well, right? But anyway, David Boaz, one of the founding scientists of this field, has served as the treasurer since the beginning. And David is here. And David, thank you very much, from every language, from every country that is here. Similarly, Martin Wolf, and you've probably all received many, many emails from Martin over the years. He is our secretary. He's the person that is probably most qualified to herd cats. And that's what it's like probably running this organization, but he keeps us all intact. Stacy Ledoux runs everything. And she has for the last 4 years managed getting this society together for this meeting. Thank you, Stacy. And and Claire Elwell. And Claire is known to all of you, I am sure. She was our founding president. She's one of our leading scientists. And were it not for Claire, that we would not have the society that we have today. And so thank you all. This is the beginning of a really, really important session for all of us. Okay, so here we go. And I want to start with a timeline here. And my pointer doesn't work very well, so I'll just point a lot. I mean, the pointer works fine. It's just that we don't see it on the screen. The timeline is from 1977 to current time. And the graph is showing the number of fNIRS publications over the years. And of course, what you notice is the rapid acceleration in the last decade. And that is in no small part due to the fact that our society began about 2012. And that acceleration corresponds with the various meetings that we've had. In 2010, we had the London meeting, then we had the Montreal meeting, and then there was the Paris meeting, and then there was the Tokyo meeting, and now there is the Boston meeting. And it was, of course, but I got it right, didn't I? So so corresponding with those meetings is the correspondence with the acceleration of the rate of publications, which really marks the influence that we have had as scientists and engineers who are studying physiological properties studying physiological mechanisms using optical properties. Okay, so just a little bit of history here, which is covered quite beautifully in Marco Ferrara and Valentina Charisma's well-known historical publication in Neuroimage a few years ago. But up until 1992, we actually knew a great deal thanks to the work of Yobsis and laboratories like David Delphi's laboratory that discovered principles for measuring brain oxygenation and could use that in clinical applications. In 1992, there was like lightning struck. It was an amazing year for what we know of now as the hemodynamic response function. That was discovered in using optical imaging by Britton Chance and his group. And the discovery was that as neural activity progressed in the brain, we could detect it with optical methods. Big discovery. But he wasn't the only one that discovered that. In 1992, there was an engineer and scientist at Bell Laboratories, Seiji Ogawa. And Seiji Ogawa made exactly the same discovery, except he did it with magnetic resonance. He discovered, now his first discovery was not with humans, it was with rats that he had in his MR scanner. But he discovered that when the lights were on, the signal in the back of the brain of his rats was greater. And he reasoned that what he was doing was imaging the responses to neural activity. So in 1992, Seiji Ogawa discovered magnetic resonance imaging could detect the hemodynamic response function in the human brain. Look at that curve. That blue curve shows the number of publications that Google Scholar reports with the search of MRI and human. It was an explosive increment in numbers of papers as we began to discover how the brain actually worked using this signal. This was like manna from heaven for a neuroscientist, because for the first time, we were able to actually look at the human brain respond non-invasively in a scanner with the anatomy and watch visual system work, the cognitive system work, the memory systems work, the whole brain working. A natural, normal human brain. We could also watch it work in cases of clinical applications in disease. But this was an explosive moment for neuroscience. Now, the principle is that we have one physiological process that is the hemodynamic response function, what happens is that active neural tissue in the brain, when it's responding to a stimulus or working, actually calls oxygen. And as it calls oxygen, it changes as it calls oxygen, it changes the amount of oxygen in the local area that's actually being activated. And so we get a signal that looks like this. And this was a signal that was imagined. It was reported by Marco Ferrari. It was imagined for near-infrared spectroscopy. One would see an increase in the oxygen, the red line, a decrease in the deoxygen hemoglobin, that is the blue line, and of course the total, which is calculated, is slightly lower. The gray bar represents the activity. So you can imagine in that 10 seconds of activity, one might do a finger thumb tapping task, and that finger thumb tapping task would respond by eliciting activity associated with the recruitment of oxygen and the decrease in deoxyhemoglobin. Okay, that is that signal as it is detected by optical measures. The same signal, of course, can be detected by magnetic resonance imaging. And here I show a seminal piece of work by Ted Hubbard. Ted, are you out here? Ted here? Ted was going to be at this meeting. Ted did this work a long, long time ago. I think it was his doctoral dissertation. He did it with you guys, actually. But what he actually showed was that the hemodynamic response function, when it was measured both by magnetic resonance imaging, which is the black line there, and by near-infrared spectroscopy, produced essentially the same signal. So here we have a physiological response of the brain, the hemodynamic response function, responding to any activity that the brain is doing, and we have two ways of detecting it. We can detect it with near-infrared spectroscopy. We can detect it with magnetic resonance imaging. And we, as a society, have spent a great deal of time focusing on this signal, how it differs, how it's the same, how we use it, how we process it, how we analyze it. That's what we do. But that's not what I'm going to talk about. I'm going to talk about how we use it to understand how the brain works. So this is my way of representing the explosive impact that functional magnetic resonance imaging had on understanding the brain and neuroscience. And this was over approximately 2 decades of the brain. And in addition to the opportunities that we had from the incredible install base of scanners that was already distributed all over the United States and all over the world. We put somebody in the scanner. There was a scanner in every medical center, and we could actually do this non-invasive imaging of the brain. So it was a situation that was just already existing, ready for this explosion to happen. And then we had the brain initiative in 2013, and I'll just read this because I think this is so beautiful. President Barack Obama announced the grand challenge, and probably a lot of you remember that. The grand challenge was to accelerate the development and application of new technologies that will enable researchers to produce dynamic pictures of the brain that show individual brain cells and complex neural circuits that interact at the speed of thought. Nice idea. And there was a lot of funding that promoted this understanding of brain coupling with behavior. The initial brain initiative started in 2013, and it was a 10-year plan. You can see it's almost over, but it's not over. It's continuing in many other forms. Okay, so we all know that as wonderful as functional MRI is, it's not so wonderful. First of all, it's a confining environment. And look, these are pictures from my scanner when I was at Columbia. Look what you have to do. You have to put a single person in the bore. you put this thing over their head that we called the bird cage, that plastic thing over there you put over the head. There's a little mirror there they can look out. We tell them that they have to hold absolutely still, because if they don't hold still, we don't get good images. And then sometimes we can tweak it up by putting apparatus on, like response buttons and maybe earphones and things. But in a high magnetic field, most of these things are the apparatus. One is dangerous if it's not magnetically inert, and two, it breaks after about 3 months because there's very little equipment that can stand the tolerance of the high magnetic field. So magnetic resonance imaging restricts our understanding of the human brain to single brains. Un problema. Okay, well, some people tried to solve that problem. I know. And they actually got funding to do that too. But there have been others that have been successful. Reed Montague, some time ago, chained 2 scanners together. And what you can do, and there's a group, a Professor Sarato's group in Japan who does this quite successfully, actually. We can have people in separate scanners doing things like playing chess and interacting in a limited way. But note, there is not much interaction of the kind that we know, the face-to-face interaction where you look at your partner's eyes and face and make inferences about what's going on in life. You can't do that in a scanner, even though there's some attempts to make that happen. So, enter near-infrared spectroscopy. So, we are, as human beings, profoundly social. We spend our lives looking at each other, talking to each other, sharing things with each other. From the moment we are born to the moment that we die, we are social. We don't need language even to be social. Look at these beautiful kids. They don't need language, whatever's going on there. We care for each other. We send special messages with facial expressions. We are social. And yet, we as neuroscientists know so little about the neural circuitry, about what makes us social, that it's a gapping, it's a knowledge gap, as we say in the language of grant applications. It is a knowledge gap that is urgent. We need to understand the neural systems that underlie this most precious, this most important behavior that we have, and that is what makes us social. So what do we do? Well, of course, you guys know the story. We turn to functional near-infrared spectroscopy. Woo-wee! And so here we are. This is September 25th, 2014, and I had my little BlackBerry at the time, and I went around taking pictures of the boxes in my laboratory. Now, this is when our equipment finally arrived for us to start doing near-infrared spectroscopy for the purpose of trying to set up algorithms and systems that actually could image the human brain in action. So I present this to you not just to give you some history of my laboratory, but this is kind of an analogy that we're starting a new science. And part of my lecture, the way I'm thinking about this, is I'm going to kind of take you through the unpacking of the boxes as we begin to think about how we can apply near-infrared spectroscopy and 2-person neuroimaging to understand how it is that our social systems operate. So this was just the beginning. We received our machines. They made an incredible mess in the lab. We began to figure out how to use it. And like good scientists, the first thing that we would do is try to assure ourselves that we had signal. So finger thumb tapping is the cardinal task for identifying signal. And so one of my students set up to do a finger thumb tapping task. Now, for all of you who've worked with the hemodynamic response function, you know that's kind of a slow, kludgy function. And so to study it, you have to have periods, or we call blocks of activity. So you have people do something for like 15 seconds, and then you rest while the signal falls, and then you do something again for another 15 seconds, and then you rest, and so on. So if you're doing finger thumb tapping, you do finger thumb tapping for like 15 seconds, you rest, you do finger thumb tapping again for 15 seconds, and you rest. And if you have your optode, your detector, right over the expected motor strip, then you would expect a signal. This is the oxyhemoglobin signal coming from our near-infrared spectroscopy system. So the first 15 seconds tapping, the signal goes up, resting, it goes down, tapping, it goes up, resting, it goes down, and so on. A well-behaved signal. We also collect the deoxyhemoglobin signal, tapping, it goes down, resting, it goes up, and it is as well behaved as the signal can be, and we can recapitulate what we knew we would expect from a well-behaved hemodynamic response function as collected both in MRI and also in functional magnetic resonance imaging. So then we begin to set up how we would do the paradigms with two-person neuroimaging. So we did a lot of practice, and finally got it set up. And so this was really sort of the turning point from going from functional MRI to functional NIRS, and it really was a journey going from single brain studies to, in this case, dual brain studies. Now, we weren't the only ones to have this idea. Felix Skolkman and Holper, Wolf and Wolf had this idea around 2013. Babolini and Astellini had these ideas. There was a lot of people thinking about let's do it, but it wasn't quite so easy to actually make it happen. There's some really nice features about functional near-infrared spectroscopy. One, we have extraordinarily high temporal resolution. We're acquiring data at about 30 milliseconds, which is amazing for doing functional connectivity studies, which I'm sure some of you do. Our resolution is not so good, where we work at about 3 centimeters across the brain, and of course our depth doesn't go any more than that. And so we don't ever image from structures that are below the surface of the cortex. So that's a major limitation. But we have no magnetic field. That is like manna from heaven. It's like amazing to do this kind of work without a magnetic field. And of course, minimal tolerance to motion. So people can sit up across from each other, as illustrated here in a situation where 2 people are participating, it looks like in a language task. One of the nice things about this type of imaging also is that easily we can combine other modalities. And so we can look at functional interactions from the point of view of the brain, but also from the point of view of the eyes, so we can measure eye tracking, from the point of view of EEG, so we can do electroencephalography. We have scene cameras, we can watch people's faces, we can do facial classification, behavioral measures, physiological measures, and so on. And there's no reason in the world why any of those secondary pieces of data are contraindicated, because we just have to pile them on. You can see here in the apparatus that we have set up here, there's a lot of additional pieces of equipment that are piled on. Okay, let's go back to Brain 101. This will be the first chapter of your neuroscience book when you were a freshman in college. And it sort of represents what every person on the street knows about the brain, and it's illustrated here with this cover of Time magazine. That is the real estate principle, that specific parts of the brain do specific things. Now, we, of course, this wouldn't be in your neuroscience textbook, but this one would, because other people have gone around and identified the functional neuroanatomy. Back of the brain is vision, hearing is the superior temporal lobe, language, sensation, movement, and so on. And so different parts of the brain do specific things. And so our job first to do the two-person neuroscience was to validate it. If we worked in the two-person mode, could we actually recapitulate or reproduce what we actually know is true about the brain and what we could do easily in the scanner? So in the scanner, we were really good at mapping the language areas, for example. So someone would go in the scanner and we would show them pictures and ask them to silently in their heads. Now remember, people can't talk in the scanner because of the movement. It contraindicates the quality of the image. So they can't talk. But if you ask people just to think the names of these pictures, you can actually map the language areas. And so we would do this regularly. In fact, we could do this with such precision that we could give these images to surgeons who could use maps for individual patients for planning resections around tumors that would avoid the language areas. So this mapping was perfected to a very high level and use. So we knew the right answer. So we started the two-person neural studies with a task that we knew the answer of. And so we set up this paradigm where two people would sit across from each other and they would talk and listen in a programmed manner, as you can see here. And then we would, they would do exactly the same thing in the scanner. What caught my attention, I wanted to remind you, this was not face-to-face. And whenever we have people doing things face-to-face, it's a whole different thing. So with this talking and listening without face-to-face interaction. And the question was, could we recapitulate in this two-person neural situation what we knew had to be true about the language mapping in the human brain? And indeed, you can see that we did this quite brilliantly. There's the map on the far right, and then the results of a study doing exactly talking and listening, a two-person paradigm. We see Wernicke's area in blue. That was the receptive area. The talking is represented in red. And we see the articulatory system, what's oftentimes called as Broca's area, speech production, Wernicke's area, speech perception. And I'm sure most of you know that. If this was replicated very beautifully in the two-person paradigm. So that sort of was the beginning of something that was, we thought, okay, we're kind of getting our wings now. We can begin to figure this out. What goes on in a two-person neural paradigm that doesn't go on in a single-person neural paradigm? And we had a hypothesis called the interactive brain hypothesis from the philosophers, actually, DePaulo and Jaeger, and actually Leonard Schielbach around the same time were writing theoretical propositions saying that, well, people's brains should be different in interaction than they are in solo mode. And it was hypothesized that it was so, but there was no data to suggest that. So we thought, okay, this is the way to start this new science. We'll just look and test this hypothesis. Is the brain different in interaction than it is in solo mode? And let's go back to our language task. And so we do the talking and listening with interaction, which means you pay attention to what the person you're talking to is saying, and you even comment on it, or you can do the talking and listening with the picture naming without any interaction. And indeed, the answer is consistent with the interactive brain hypothesis. Wernicke's area on the far right in blue is much more exaggerated and larger during the interaction than not in the interactive case. It doesn't, apparently it's kind of funny, apparently Broca's area, the parts of the brain that have to do with speech production don't care. Could care whether you're talking to yourself or your neighbor or anybody. It doesn't make any difference. Talking is talking, according to the brain. But if you're listening to somebody else and paying attention in interactive mode, it is the back of the brain, that temporal parietal junction back there, that seems to matter. So data are consistent with the interactive brain hypothesis. So we say, okay, if that's so in language, is it true in vision? Now we know a lot about face processing. Faces are probably the most important social stimulus that we have. And people in MRI for the last 2 decades have studied face processing. And so we needed to ask, is it matter that you're looking at a real face, or is it just a face? And that's what the face processing models currently suggest, that faces of all kinds occupy similar processing mechanisms. We ask the question, if the face is real, is it different from the face that is simulated? In this case, we use picture faces, we use video faces, and in some cases we even used robot faces. And the answer was always the same. It mattered a lot. You couldn't fool the human brain no matter how hard you tried. A real face was a real face, and if you didn't give the brain a real face, it knew it. And it mattered a lot. This activity in the red spot over here in the right temporal parietal junction has been known as an area that was active in social conditions. Oh God. Jeez. All right, I'll speed it up. We got 30 more minutes? No? Oh, okay. Phew. All right. All right. Okay. So it matters a lot that the brain that the face is real, and this was again a real. This was a turning point for us actually, because we realized that this was the point at which that we probably had a whole new area of neuroscience ahead of us. That we knew that live interactions influence brain processes, and that we had both the technology and the mathematical capabilities to actually study it. And so it was this background that really encouraged us to start thinking that the neuroimaging of two brains simultaneously was going to be a fruitful area of investigation. So I'm going to take you down a road a little bit about how we go about that. Now, this is a little deeper dive, and I'm going to warn you just a little bit. There'll be a couple of data slides, and I won't have all the answers. It won't be quite as clean, quite as neat, but it's neat science. So hang on, it won't take too long. But we want to know what in the world those circuits are doing. Why? What are they doing? How does it work? And it's common knowledge. One does not have to go back to Cicero in 45 or 46 BC. But nonetheless, I'm making the point here that it has been common knowledge for a long time before there were neuroscientists, that the face and the eyes were very important for socialization, communication between individuals. And so I love this quote by Cicero: "The face is the image of the mind, and the eyes are its interpreter." And I thought, aha, this is the beginning of a hypothesis. The hypothesis is that eyes, and meaning eye-to-eye contact, has access to the brain, and the faces are the source of social information. Well, those are nice words, but we could actually put that into an experimental paradigm. So, eyes. We can measure eye-to-eye contact in our two-person paradigm by setting up our eye trackers. And we have an eye tracker system in our computers that tell us when each person in the partnership has their gaze focused in the eye box of their partner. And when it stays in that position for 10 frames, which is 0.8 seconds, we say we have an eye contact event, and we can collect these eye contact events. Now, while this is going on, of course, we can do the neuroimaging because we have optodes on the heads of our participants. And I want to just review this this paradigm down here for neuroimaging because it's a timing paradigm. The three blue stripes represent 15 seconds of activation, and then the blank area, 15 seconds of rest. So remember the finger thumb tapping paradigm. You tap for 15 seconds and then you rest. You tap for 15 seconds and then you rest. It's the same hemodynamic signal, and so we have to treat it exactly the same way. So we have 15 seconds of resting, 15 seconds of action, and then resting, action, resting, action, resting. But why do we have those three bars? Well, because those three bars are 3-second moments in time, and it's because we're asking people to look at each other Now, have you ever tried to look at somebody for more than 3 seconds? Doesn't work. You can't do it. People can't do it. Not even our Yale undergraduates. They look at each other and then they start to laugh, or they look away. People cannot look at each other for more than about 3 seconds, and they look away. And so we said, okay, fine, fair enough. So we set up this 3 seconds on, 3 seconds off, 3 seconds on, 3 seconds off paradigm, even though we analyze it all together in many of our paradigms. But all of our face-to-face experiments, and particularly those where we're measuring eye contact, we do 3 seconds on, 3 seconds off, 3 seconds on, 3 seconds off. Try it. Just do a little experiment. Try to stare at somebody for more than 3 seconds and see what happens. It's very hard. Oh, see, some of you guys can do it. Okay. Well, anyway, our Yale undergraduates couldn't do this very well. And so this is a standard paradigm that we set up. So we've got two things going on here. We're measuring eye contact and we're measuring the brain responses to face processing. And remember our hypothesis. Our hypothesis was that the eye contact actually would affect the brain processing. So what we did was we took the eye contact data and we used it as a covariate. We used it as a regressor on the neural activity. So we modulated the neural activity by the eye contact. And so what we see is an amazing neural system here that represents eye contact as a covariate with face gaze on the neural data representing face gaze. So here, the truth is, Cicero was right, that eyes and brain have a very special relationship, and that special relationship can be actually quantified and observed in what we like to think of now as a dorsal or dorsal parietal region. That top of the brain on the right side, in the sort of the sweet spot at the top of the dorsal parietal lobe, somatosensory association cortex, the supramarginal gyrus, that region has been known by others as being a region that's associated with salience detection, movement of the eyes, for example, to important topics, important subjects. And so there's a lot of face validity in identifying that area as an area that was specific for faces and eye contact. This is a system that also involves the dorsolateral prefrontal cortex, and I want to save my comments about the front of the lobe, and I want to stay in the back of the lobe for a couple minutes here, because we want to go on and say, okay, we've got this system, but what does it do? What does it do? What does it mean that we've got this system that's associated with eye contact and faces? That's pretty cool, but it's not there just to look at eye contact and faces. It probably does something really important in our social lives. Well, we hypothesized that it probably had something to do with social communication, that the brain uses this information in a dynamic way. And this is one of the things about the kind of work that we're doing now. It didn't really get at the dynamic use of the eye and face system. So we test this hypothesis with a new kind of paradigm. And this paradigm is sort of unusual. And this is the other thing about this type of work, is that we didn't have built-in paradigms. I've never done science before where somebody hadn't gone ahead of me and sort of written the recipe for how you do the experiment. There was no suggestion box. There was no instructions. No one told us how to do this social neuroscience stuff. So this is something that we just made up. So we have one person that we call him the movie watcher, and we give him interesting material, which I will share with you in a minute. This one person is a stimulus. And then we have the person on the other side of the table. We call this person the face watcher. And the face watcher only watches the face of the movie watcher, and the movie watcher is watching the movie. Okay? Very cool. So this is our dyad, face and movie watchers. Okay, so now what do we give the movie watcher? Remember, the only thing the movie watcher has to do is give us a facial expression, really. That's what we're interested in. So we give him a category of stimuli we call the adorables. And these are like 3-second little vignettes of things that you just can't help but smile. And no matter how many times you see them, you smile. We call them the adorables. And then, guess what? We have another category that we call the creepies. And you can imagine what the creepies are. Golly, creepies. Okay, they're kind of worms and spiders and nothing violent, nothing that anybody else has ever used before. We pull them off the web. They're just things that make you go, ew. And then we have our controls, the landscapes. Okay, so we got these three kinds of things that we're giving the movie watcher, and he's responding naturally. Okay, so the situation is kind of like it's shown over there on the other side. We have a what we call a smart glass that we put in front of our participants. And the smart glass, if you see down on the time series below, the blue bars, we have 15 seconds of watching. The glass is open, and then 15 seconds the glass is closed. Glass is open, glass is closed, glass is open, glass is closed. And so people can have their rest period, which we have to have for the hemodynamic response function. Now, during the watching, the movie watcher is watching the movie, never watches the face of the face watcher, but the face watcher is always watching the face of the movie watcher. Now, during the 15-second rest period, each of these participants does a rating, and they rate how their stimulus makes them feel. The movie watcher rates how happy or unhappy or positive or negative the little vignettes make him feel. And the face watcher rates how happy or otherwise the face of the movie watcher is. So they're doing the same rating, but with different stimuli. Okay, and then we have one other thing. We actually are able to classify faces by their facial expressions because we can take advantage of action units that have been, this is a computer platform that has been with us for several years, 2016 actually, that is designed to quantify facial expressions. So it quantifies facial expressions by the positions of the facial muscles. The articulation of the muscles of the human face is very precise. And there are numbers of them, more than what we're showing here, of different classifications. And so this program goes through, watches the face of both the movie watcher and the face watcher, and identifies the facial expression at any given time in the time series. So we have three variables here. We've got neural responses to face gaze. That's the face watcher watching the movie watcher. We have facial expressions of the movie watcher, and we have the affect ratings of both of them. Okay, so what do we do with that? Well, the first thing we want to know is, have we been successful at creating a social situation? Remember, these people aren't really looking at each other, which is very odd for a social situation. But when we correlate the ratings of how they are feeling, you can see a very high correlation between the ratings of the movie watcher and the ratings of the face watcher. And you see that it's very consistent. The adorables are rated at the high end of the screen. The creepies in blue are rated at the low end of the screen, and the neutrals are rated more or less in the middle. Notice in the blues, the spread in the action. That was because, again, it's Yale undergraduates. One person across the screen goes, "Ew," and the other guy laughs at them. That's what that spread is. They don't all do that, but that's the spread of the data there. But anyway, so we consider this a kind of a demonstration of emotional contagion, if you will. And so when we do something kind of interesting, I think, we take the ratings from the movie watcher, That is how he felt watching his movies. And we use those ratings as a regressor on the brain activation of the face watcher. We have an imprint, essentially, of that person's emotional feeling on the brain of the face watcher. So this should be a neural system that actually defines, say, emotional contagion. And here it is. It's a pretty nice system, actually. Again, it is heavily weighted toward the dorsal aspect of the parietal lobe. It has, of course, a bit of the on the right-hand side, a bit of the visual system as well. We see a lot more left-sided activity. Their left side is the brain on the far right over there. Again, because we do see, and I can't explain why, but this has been known for a long time, we do see quite a bit of lateralization of these effects with a bias toward the right side, which is oftentimes why I show you only the data on the right side. Here, this is a little bit different. One would think, based on what we know from functional MRI, that that system on the left is more of an object processing system, but it does many things. It could also be an auditory system, but there's nothing auditory going on. So I don't understand why we're seeing activity on the left, but we would have predicted what we're seeing here on the right hemisphere, the left brain. We can do the same thing with the facial expressions. We take the facial expression of the movie watcher and modulate the neural activity of the face watcher. So we get the face of the movie watcher embedded in the neural activity of the face watcher. Again, we see very interesting dorsal parietal systems. Now, this is work that's ongoing. We're going to go after this system with TMS to understand more about what this system does. So I told you this was kind of real science. I haven't wrapped this up, but we think that these systems here are extremely important in understanding social interactions on an active basis between two individuals. So just trying to wrap this up, and I want to go on to another topic, but what we've learned here from this is that this dorsal parietal system processes social cues, and that it's become a really important region of interest. We didn't know about this system before we started doing this work. These systems are also relevant, we think, to clinical conditions such as autism, where social interactions of this kind are very much a hallmark of the disorder. And we have some preliminary data suggesting that indeed it's those systems that are affected. So I think we're going to hear a lot more in the future about these types of systems, these types of paradigms. Now, I want to unpack another box a little bit. I want to just go up a menu, and I want to leave that line of work for future directions and ask a little bit bigger question about does the dyad really matter? That is, we go back to the interactive brain hypothesis. Are your brains really different when you're in different situations with different dyads? And so we have several examples of this, which I want to share with you. But again, in the spirit of perhaps laying some groundwork for future work that many of you might actually do in the future. But in the first case, dyadic interactions, we all know the interaction caretaker versus patient. Well, is that a different neural system than just talking to somebody in a more neutral situation? And the answer is, when we do this study, that it is the frontal lobe that comes through as the part of our brain that seems to be distinguished, specifically activated during conditions of caregiving. When you're in a situation where you're actually with a patient, with providing advice, the frontal lobe is the frontal lobe that is providing the additional information above and beyond all the expected systems that you would expect to be acting, including the visual systems, face processing, and the language systems. So just hang on to that thought. Another situation, imagine that you are on a bus and somebody sits next to you who is clearly from a very different socioeconomic world than you are from. And you, being who you are, strike up a pro-social conversation with that person. Is your brain different in that situation than if the person who had sat next to you was one of your college classmates or one of your business associates? There is no theory out there that says that there would be any different other than the interactive brain hypothesis. And until this work, there really hasn't been very much evidence that your brain is different in this challenge situation. And indeed it is. The frontal lobe, again, is the part of the brain that appears to be most active when you are in a situation you are challenged to provide pro-social conversations with others. Finally, do you think your brain is any different when you're agreeing with somebody than when you're disagreeing with them? Woo! I bet you could guess this one, right? Okay. We did this situation where people were in an agreement situation and a disagreement situation. These were views that they held closely, naturally. They weren't views that were made up. The red and yellow are the brains in disagreement and the blue in agreement. Notice the blue, the supramarginal gyrus, the right side social areas, are most active in agreement, and frontal lobe profoundly active in disagreement. Again, making the point that what's going on in a dyad really matters. Now, what's wrong with what I told you is that I haven't taken it far enough. We don't want to just be doing gee whiz research, finding after finding. What we want is a theoretical framework that provides for us falsifiable hypotheses. And we're not quite there yet. This is quite a new field, but it's work like this, I think, that provides us kind of a grounding. And I have one more kind of area that I want to touch base with before I wrap this up. Again, it is a new area. And this is the point that I kind of want to make throughout this, that this is just the beginning for this new kind of science. And what we're providing is some kind of neat stuff to get started with, some beginning training wheels, but we've got a long ways to go yet. And this is another one of the parts of two-person neuroscience that I think is very, very exciting, a neural coupling. When two brains are interacting, what we see is that the neural responses are corresponding. They become synchronous, which is really interesting. This was a phenomena that was observed by Cui at Stanford using functional near-infrared spectroscopy in 2012, and it follows up on ideas by Yuri Hassan and Frith and others that were working with MRI, who proposed that when two brains were synchronous, they were actually sharing information, which I think is an extraordinarily interesting hypothesis, partly because it's testable, and partly because it makes a lot of sense. But anyway, this is the neural coupling is the extent to which neural responses across brains are synchronous. That is its definition. And basically what happens is that you have responses from one brain, and then you have responses from another brain. The extent to which the temporal courses of those responses are in sync is the extent to which they are considered neural coupling. And we oftentimes use wavelet analysis for this. Now, there's many, many examples out there of neural coupling, and I'm sure that a lot of you have seen them. It seems like it's very common to observe that when two parts of the brains from two different individuals are active in a socially engaged manner, that there is a synchrony between the systems. And it is, I think, what I think is one of the frontiers in front of us, these neural linkages. And I don't know exactly what they're due to or how we can manipulate them. My hypothesis is that this is where the sensory systems link to the brain, and probably is the synchrony is related to visual sensing, or how the eyes are moving, or synchronous or reciprocal motions of the face. But this is a very active area of research. And so again, I leave this as a job to be completed with future investigations. So what's the news? First, I think we can say we've done a little bit. We've established that dyadic interactions are a fundamental unit of behavior. And I don't think that statement could have been made, at least with documentation, much before all of us started doing the two-person neuroscience. Secondly, that this work has introduced another whole genre of experimental paradigms, computational techniques, and measurement tricks with many different modalities to actually accomplish the understanding of what goes on in two people's interactions. Third, we learned that, and this isn't too surprising, but the principle of neurospecificity that applied to single brains applies to two brains as well. Brain works with very specific activity, very specific areas for very specific tasks, very specific systems. So the principle goes on even in two-person neuroscience. And then finally, and I have the least to say about this, although we always publish this with our papers, about the neural coupling as being a property of interacting brains that we hope to understand better in the future. So, the future. This is my crystal ball. You like my crystal ball if there are two brains? What is going to happen in the next few years? So we go back to my timeline, right? I like my timeline. And here I notice I'm using a log scale this time now to show the numbers of publications. And that's so I can put fMRI on the same scale as fNIRS. So I've got 3 lines up here. And there's fMRI, a number of publications over the years. And just note, don't laugh, but just note, it's kind of leveled off, you know, kind of just leveled off. But note the fNIRS line. It's accelerating. And that means that, the impact of the work that is being done by optical systems to measure physiological processes is making a impact impact. But now, the third one, fNIRS hyperscanning. Now, hyperscanning is not a word that I've used very much, but it's a common word that implies two brains, two persons. And so if you wanted to know kind of what the status of the field was, the search terms would be human, fNIRS, and hyperscanning. And that's what that green line is. Those are the search terms. Notice that that one is accelerating at a rapid rate as well. So that means if past performance is the best predictor of future performance, that means we've got a pretty cool future ahead of us because there's lots of action, lots of things happening. So let's guess about what that might be. Well, first, I think that not too far down the road, that two-person neuroscience, as it is measured by near-infrared spectroscopy, is going to be a mainstream science. We're not mainstream yet, but I bet you that we will be. Another 5 years, we'll have our own meeting, maybe even. Imagine that. But it's going to be a mainstream science. 2, that this mainstream science is going to have a theoretical framework. We're not just going to have the interactive brain hypothesis. We're going to have some pretty well-developed theoretical frameworks about what those networks actually do and how. So there's a long ways to go there. That we will have clinical applications that give us insight into disorders that disorders with social dysfunction. For example, autism, some cases schizophrenia, other cases depression, for example, when social interactions are a hallmark of the condition. Let's hope we have solid funding sources. But I think NIH is listening. They better start if they're not. NIH, NSF, Wellcome Trust, Gates Foundation, whatever. This is going to be a field that people are going to have to take note of. And hopefully we can all work together. And I like what Maury said early on in her introductory comments. We all work together to make sure that we can secure funding for this work, not only this work, but for all of the work that we're doing in brain imaging. We got to keep that brain initiative going forward. I expect that we will have continued development of the technological infrastructure. The fact that we are an organization of engineers to build widgets. You guys build equipment. And if you didn't build the equipment, we wouldn't be able to make the discoveries. Discoveries depend upon equipment and ways of measuring it. And so we are, as scientists, forever indebted to you who build the engineering that does the detecting for us. And finally, I think we need a journal. I think we need a journal sometime in the future. I see some heads nodding here, right? Okay, you guys, I'm appointing you, editors, right away. Okay, got it. We need a journal that represents the two-person neuroscience, the journal that reports and archives the developments that I expect that we're going to see in the future, hopefully. Okay, now we are standing on very solid ground. There was this very impressive issue of neurophotonics. It just came out, I think, last month. It represents the 2022 status report on where we are. And we have to thank Rickson Mesquita and Aaron Buckley for putting this out. There are 59 sections, a whole book of neurophotonics. And it really spends an awful lot of time on hardware development, data analysis and algorithms, the functional applications of neural development and cognition, and clinical applications. Now, we who do two-person neuroscience got a couple of little paragraphs in that. Couple of paragraphs. And thank you, Rickson, for that. But there's going to be more next time, believe me. But the point here is that we are standing on the shoulders of giants that have built the infrastructure to make this happen. And because it is there, because this whole hardware development, data analysis on is there, we will be able to take it forward and move it into the future. And so with that, just 1 second here. I want to read something, so this will help me do this a little bit better. I want to, this is my standing on the shoulders of giants slide, but this is my last slide. But I have a couple of things I want to say. That, like any laboratory, my lab is, it's a lab that does all the work. In a lab like mine and like all of your labs, there are people that are smarter than you, they do more than you, and they take the science forward. And so I have some credits that I wanted to give to my lab for doing all the work. And the credits come in the form of the work that is actually being done here, because these future directions that I talk about, they're not just pie in the sky. This is work that is really happening. And so I'd like to call attention just for a list, a few things that you will see at this conference that represent future directions of the kind that we've talked about here. And the first is Adam Noah's poster looking at TMS and going after that dorsal parietal network, going to figure out what it does. That Chen Zhang is working with machine learning to diagnose autism using facial expressions with autistic individuals. Tamara Parker is actually using eye gaze. She's giving a talk here to look at the local areas that in face processing in autism that are defective. Megan Kelly is looking at the temporal resolutions of face processing in order to identify basic properties of visual sensing in face processing in a two-person dyadic arrangement. AZ Alsop and Amanda Watts are looking at the role of face processing and music as a possible way to be an assist to music therapy. Mark Tide is looking at the interactions of social and language systems during language encoding. Uz Hakim is a graduate student that works between Ilias Tuxidis and my labs at UCL, has developed this new system of hyperscanning across laboratories. So it's the neatest thing in the world. We have one hyperscanning team at UCL and a hyperscanning team at Yale, and we're doing science. We had so much fun setting up the communication program protocols between the two labs. We almost forgot to do the science. But there's some science there, so Uz's poster will present that. Sarah DeFelice is studying social learning and dyadic interaction, and Nan Zhao is somebody that has looked at interactions in real life as opposed to Zoom. And so you want to hear those. So the field of optical imaging, I think, for two brains has a very, very bright future. Thank you. Let's see.
Yeah, thank you very much for your great talk. It was awesome. And well, the time is limited, but we would like to accept a few questions from the audience. Do you have any? Question? Okay.
The microphones are there.
Yes, it's there.
So
You want to move the-
Yeah, okay.
Go ahead.
So psychology has some of the universally recognized emotions and facial expressions. Are you potentially considering using different dyads of examining how facial expression is analyzed between different regions, say the United States and Europe and other countries?
That's such an interesting question. The question is about the cultural impact of a facial expression and how a facial expression in one culture has a different interpretation in another. It's a fascinating subject. And I'm more concerned at the moment of having facial expressions of any kind impact a brain in any way rather than the specific facial expression. But it's a really important point about how the language of faces is different in different cultures. And I'm sure if we put our heads together, we would find a way to study that. The answer, I think, is going to be pretty granular. And I'm not sure that we have the resolution to go there. There might be something that we might have to take to the scanner. Once we understand how to do it, I'm perfectly happy to go back to the scanner and do studies where we can actually refine the resolution of our results. But it's a very interesting question. Thank you.
Thank you. Next one, please.
Thank you. Dr. Hirsch, thank you for such an illuminating talk. Your lab has produced a lot of diverse hyperscanning research, but I was curious as to what prompted your sort of shift from fMRI into fNIRS.
Oh, okay. A really interesting question. Thank you for asking it. I had done fMRI for quite a while. I loved it. But I was really bothered by the fact that the questions that I was really interested in, that my research had led me to ask, weren't being answered. It was really the question that moved me from this single brain paradigm to the double brain paradigm. I think that the science is so much richer, so much more interesting, so much more fun, and probably so much more important when we can study two people together. I also really like the idea of moving toward environments that are more free acting. When I have to put somebody in a bore and tell them that they can't move, and then I put them in an environment with like 110 decibels of banging, and I ask them to do things, I think it's a little restricting, don't you think? And so it was just so much more fun to imagine that we might be able to learn more about things that were more important if we just went to another technology. Thank you for that question.
Let's have just a final question. It's okay?
There will be a reception afterwards, and I'm happy to ask questions, to answer them. I'll ask them too.
Hi, thanks for that amazing talk. It's always great to see your lab and all the technological advancements that you guys are working on. My question is, I guess, have you ever thought about linking back to the developmental literature? Like, as I was looking at what you were doing, there's so much interesting work about mothers and babies and all that kind of stuff. So like, could we bring these fields together?
Oh, that's such a good idea. There is so much more to do out there. And yes, I mean, it's a natural to go back to the developmental field. And there are some of you out here that are actually doing the mother-child-infant studies. That's so important. None of it's as easy as it sounds. But I think that some of the most important questions in the two-person neuroscience arena have to do with development, because face processing is probably one of the most important but first things that we develop around, and understanding how that affects our social behaviors be really important. So developing ways to study the infant adult dyad is really important, and I know that there are people that are actually working on that. I'm very humbled by this work, actually, because it's hard. It's hard to figure out how to do these things. I think we have to support each other as we make mistakes and figure it out. We'll get there. We'll get there.
Okay, thank you very much for you are raising lots of great questions, and our time is limited, and it may be a time to go to the reception, and we thank you very much.
Thank you. Thank you.
Thank you. Okay, so the reception is out and in. I don't think they will open this. And you have the yellow tickets are for 2 drinks and food as much as you want. And if it's cold, you can stay in the poster area. Thank you, everyone. Close this.